All right, as we look at the science, I just want to, this is really at the heart of what the High Wire does is an investigation into what is happening around the science of vaccinations, what's happening to our bodies, what are these things doing. I got to see a few videos recently that just blew my mind, and I've been really excited to try and get this next guest on the show. And I want to make it clear, because a lot of what we do in this investigation is get into therapies, get into ideas that are happening. Some of them are hypotheses, some of them are proven, we've got peer review, but as you've seen, science is an ever-moving space. So I want to be careful to say, in all that we do here at the High Wire, you need to do your own investigations. Don't just take it, hook, line, and sinker. Also recognize that we're involved in an investigation here that is ever moving. So some of the views that are about to be shared may or may not be views shared exactly by the High Wire or ICANN, but it's a part of our desire to try and triangulate the truth of what's really going on here, to be a part of the scientific method, challenging it from all sides. No one is bringing a more, I think, clear and riveting challenge than Dr. Sabine Hazan, and she is going to join me in a moment. This is what she looks like in the news.
Joining us is gastroenterologist Dr. Sabine Hazan.
Gastroenterologist Dr. Sabine Hazan.
Dr. Sabine Hazan is a gastroenterologist and CEO of Progenibiome. She's a gut bacteria expert.
The whole basis of my research lab is really to write the dictionary of the microbiome and disease. We are seeing that if you have an imbalance in the gut, you are more predisposed to disease.
You're basically an expert at running clinical trials, right?
Yes. Clinical trial gave me a view of what was the future. So when clinical trials didn't work, I would do a procedure called fecal transplant, taking stools from a healthy donor and putting it into the patient. And it was amazing because you would have a patient that was dying and miraculously he lived after fecal transplant. In the world of the microbiome, unfortunately, we only know about the 1%. We've only identified about 15,000 bugs, and there's about trillions out there that we have no idea what they're doing. Will humanity survive when we start introducing 4 billion prescriptions, per year given? Will humanity start surviving when we change the microbiome and remove the diversity?
You and your team were the ones that discovered COVID's entire genetic code in the stool. So why do you think the virus was found there in the first place?
COVID actually sits on the ACE2 receptor. Where is the biggest organ where the ACE2 receptor are found? The ACE2 receptors are found, the bowels. So it made sense to me to look there because that's where everything ends.
When I saw that hydroxychloroquine and Z-Pak had an impact on COVID in the stools and started the creation of my protocols and writing my protocols, I lost no one. No one died. So we're talking thousands of patients. And then all of a sudden on Twitter, a post goes off. Dr. Hazan is doing unethical protocols. So this is the patent of azithromycin, vitamin C, vitamin D, and then hydroxychloroquine.
What I understand, a lot of people have used this formula that you're saying. You're not collecting on this.
No, I'm not. It was my donation to humanity. It was like a mark in history, being on the right side of history. To me, that was more important. We're entering into a world of individualized medicine. I think doctors need to be doctors again.
It's my absolute pleasure to be joined by Dr. Sabine Hazan. Thank you so much for coming into the studio today.
Thank you for having me.
I've been watching some of these videos now, and there's been a lot of celebrity of great doctors that have stepped out. But as I start looking at the work that you've done, in many ways, some of that's sort of built on the work that you've done. And so, I'm just so honored to have you joining me.
Thank you.
Right now, of course, you did a great interview with Epoch Times, but I want to talk about just this. What caught my attention is when we were looking through some data, and someone would say, did you see this video? It was a discussion about a study you did where you took some of your peers. You knew they were about to get the COVID-19 vaccine and said, hey, could I get, what was it, fecal samples?
Or, yeah, fecal samples.
So fecal samples
what I do.
Before you get the vaccine, and
yes.
then after. Tell me what it is, what made you decide to even ask that question?
I think in order to answer that question, you kind of have to go back to the history, my history.
Okay.
So I'm a gastroenterologist, and I've been doing, I was doing GI for a long time, and then GI went from GI to clinical trials for pharmaceutical companies. Mm-hmm. And then, one of the bugs, which is known as Clostridium difficile, which is a bacteria that, kills a lot of people.
C. diff.
C. diff.
So we
yes,
hear about it in the news
yes.
a lot, yep.
So C. diff was essentially killing a lot of people. And I was known in the world of clinical trials as the queen of C. diff.
Okay.
Because pharmaceutical companies would come to me because I would recruit these patients, try them through different clinical trials, and then when the clinical trial didn't work, I would do a procedure called fecal transplant.
Okay.
Fecal transplant is essentially the process of taking stools from a healthy donor. We don't extract it, they just donate.
Yeah.
And then putting it into the colon of the person that is sick with C. diff. So the first case, and this was a movement that was started in GI, started in the 1950s. Fred Einsman was the first one that did the first 2 cases by enema.
Okay.
And then Dr. Tom Barodi, my partner, also, started it in the 80s doing a lot of cases. And writing the data, presenting it to the American College of Gastro and different meetings, and it caught on fire within the GI doctors because we started seeing something that was working, right? So when that was working, at least, even though I put patients on those clinical trials, I always knew, well, you know what, I'm gonna have the funding from the pharmaceutical company to pay for these clinical trials, right?
Yeah.
So I felt I'm putting the patient through the clinical trial, I'm doing the trial, but then I have funds to do these fecal transplants, right? When fecal transplant became a capsule of poop,
Right.
I looked at the whole field and I said, okay, so I've been doing clinical trials for pharma for at least 3 decades.
Yeah.
We have gone from antibiotics for everything
Yeah.
to biologics for everything. Remember, biologics started for Crohn's disease. Humira, Remicade, you hear it all
Right.
over the news. And then to, unfortunately, biologics for psoriasis, for everything. So then now we're in the fecal material business. That's what's coming. And I said, we're literally going to be giving people fecal material in capsules, and we have no idea what we're doing. Right?
Because just sort of kind of, which ironically, and as I've invested, I'm not a scientist, I'm just a journalist, but when you really look into the very first vaccines, when you look at the first one, all of it is just kind of this guessing game. Let's take this pus, slap it, and go, oh, it looks like it works.
Right.
And it's amazing how many, sometimes centuries, the science goes on, and no one really ever asks, why is it working? We don't really understand the mechanism, yet the science is building this foundation and saying, we know everything. You actually don't know much at all, right? And this is what is really shocking, I think, to people.
Exactly. And especially in the microbiome space, when you look at the microbiome, what is the microbiome? It's trillions of bugs in your bowels.
Trillions.
Trillions.
With a T. Okay.
Around you, in the air, on your skin, in your nose, in your bowels. We only know about 10,000 bugs. But of those 10,000 bugs, we probably know about maybe 500, and that's being generous, of what they do.
What they actually do.
What they actually do. So we have about 1% that we've identified. I'm being generous.
Okay.
And there's about 1 trillion that we have no idea what their names are, what they do, and this is just in the bacteria level.
Wow.
There's a virus level. So we've isolated a few viruses, right? Epstein-Barr, HSV, CMV, JCV.
Right.
But there's a whole world of viruses out there floating around that we have no idea what names they have, okay?
Right.
So
Which, by the way, is one of my arguments I had with a friend
Yes.
that was like screaming at me because I didn't want to use the COVID vaccine. He's like,
Right.
we could finally be safe. I was like,
Right.
I don't know how you feel safety adding a 17th vaccine onto the 16 you're already taking in the face of probably
Right.
millions of viruses you're doing nothing about. I don't know why that makes you feel safe, that this tiny little percentage, this little like rice drop in an ocean has anything to do with actual health. It's kind of crazy because pharma just tells you, get this one and then all your problems are solved. We'll worry about the other million things we don't understand at all some other time.
And that's the problem. That's exactly why I stepped into this, right?
Right.
So I went from not understanding, well, how did we fix C. diff, right? How did the process, what is in the stools?
Why
what
is it working?
microbes
Right.
are acting with what, right? So I felt that, well, and at the same time, if you remember, there was a company called Ubiome that was selling stool samples to everybody, and then other stool samples. And I looked at that and I said, well, they have a good idea. Let me send some of my stools, right? Before and after I treat C. diff. Let me see what I see. I couldn't see anything. It was basically nothing. And it was not even reproducible. In other words, I test the stools the first time, and then the same stool sample, I get a completely different result. So when that started happening, I said, well, this field of stool testing is non-validated, non-verified, and non-reproducible. That defies science and research to begin with. Because the one thing about if you're going to do an experiment in research, you got to make sure you have the same results over and over again. Otherwise, you're comparing apples and oranges, right?
Right.
So if I'm comparing your stools to mine, for example, well, we can't be compared to each other. We're from different areas. We've eaten different foods. So when you look at these stool samples and it tells you normal, well, what's a normal? Those, you know, I'm a scientist. I'm a rebel. I question the narrative constantly because it's my body, right? So I need to understand.
You mean to tell me that just, you know, whatever the consensus says is not where you like to live? You don't like living in the consensus?
I'm the opposite. In fact, I was probably a rebel in med school. I was a rebel in residency because I believe that science evolves, for one. Science changes. One day, how much, you know, in medicine we've seen so many changes over the years, right? Digoxin 1 year is great for heart disease. The next year, hold off, it's not great for heart disease, right? One treatment for a bug is important, and then the next, 10 years, it's not important, right? So it all depends who is the science of the hour, the scientist of the hour that becomes, popular with his narrative, right? I mean, and this moment could be me, right? I could be saying it right, or I may not be right? Like the disclaimer you say, right? I'm on a path to discover the microbiome. I could be right, I could be wrong. Some smart ass would be, pardon my language, will probably try to disprove me, but that's fine. I acknowledge that. I like the criticism because criticism is what allows us to look at the other side of the scenario. So from fecal transplant, started seeing a lot of improvement with fecal transplant, then started noticing that there's a lot of wrong microbiome stuff going on, in that there was no validation. So I basically opened up.
So it turned into like a fad, but the fad wasn't actually getting to
the fad had no basis.
Got it.
None whatsoever. And so I started saying, well, I think the best way to do this, to look at the microbiome, is to essentially take the microbiome at a clinical level. Let's look at what the Japanese in Japan look like versus the Japanese in America. Let's look at the vegan versus the carnivore. Let's look at what medications are doing before and after in the microbiome. And essentially, this was kind of like a path for me of discovery, of asking questions and wanting to understand the questions. So, so my path, how did I get here? COVID hit, and I've been doing clinical trials for pharma for 30 years practically. Since University of Florida, my first year of GI. And I couldn't understand why a vaccine that was first of all, I couldn't understand a virus that was mutating and giving it a vaccine for everyone, thinking that we're gonna just stop this virus, right? And then I couldn't understand, why are we stopping early treatment? We should be turning off the fire. In other words, a couple of scientists have this idea. They put this idea and they turn off the fire. A couple of scientists have this idea. They turn off the fire this way. And then that. So basically, to me, it was more important to turn off the fire quickly, save as many lives as possible, and then look back. what
I mean, we
hose-
literally, when you think of the term fire, we said in hospitals, come get tested.
Right.
Oh, as it turns out, the fuse is burned. You are now on fire, but go home. You're not totally in
Right.
flames, and it hasn't gotten out of control. When it gets out of control and you have a forest fire in your body, and it looks like you're going to lose your house, then come back here and we'll
Right.
see what we can do about it. I mean, it's the most insane thing we have ever watched in a world that has always said early treatment in every single problem there is in medicine is best. We said there will be no early treatment. You can only come back here when you are in full on,
Correct.
death,
Yeah.
defying death.
So absolutely, and so that was essentially the problem, right? Those were the red flags to me.
Yeah.
Those were the red flags that said, why do I, first of all, remember I was in 2020, in March 15, I was writing protocols. So I was studying, when COVID hit, I was still in the pharmaceutical world of, hey, clinical trials, this is the way we do it, we go with the FDA. I was still doing all that, right? So my thought was, well, this is a chance to save humanity. I'm gonna step in as a clinical trial physician. And I'm going to look at what is the best protocol that I feel is the best protocol. So I had a couple of, running hurricanes, as I like to call them. The first thing was I had created, because all these labs were non-validated that analyzed the stools, I had 4 years prior to that started my own lab to look at the genetics of the gut microbiome.
Yeah.
So I was already on this path of, let's look at the microbiome. We're heading into a world where pharmaceutical companies are going to be developing products with fecal material. I want to know, does my fecal material patient that's coming have bipolar disorder? Because then I'm swapping C. diff for bipolar disorder.
Right.
Right. So I, as the consumer, if I ever get to be the patient, would like to know who is my donor, right?
Right.
Who is my donor and what does my donor have? And and
Right,
I know.
so that's so interesting because if just taking a fecal transplant fixes this one problem, is it bringing over a problem that donor might have had? Am I taking that on?
Yes.
Is it that? I mean, it's almost like computer programming then in a way.
It's a little bit like computer programming. I mean, it's remodulating the genetics of the microbes. Yeah, so it is a little bit. So yes, I mean, we have seen cases of, first of all, we've seen cases of people dying from fecal transplants.
Okay.
So obviously, we were aiming to fix C. diff, and then we put in another bad virus or bacteria in there that killed those people. We have seen miraculous cases, when you do the analysis properly, when you look at the microbes.
You give me one image of this alopecia.
Yes,
Let's just share that, just a sense of, imagine if your microbiome could just achieve this.
Yes.
Do we have that image? It's coming. Here we go. I know I jumped way out of line here. This is what I do to my team. All right, we'll come back.
But this is the story.
They'll let me know when they have it ready.
So basically, this was the beginning for me, was, 2 cases of alopecia areata with C. diff. And we essentially, fixed the C. diff with a new microbiome. But on top of that, the side effect was they grew hair.
All right, here we have it. I think we have the image now. Look at this, folks. Just simply by doing, this fecal transplant, you fix the C. diff, but look what happens to the hair. I mean, this isn't totally shocking. People have been watching our show for many years, but if you're new to it, this is really, I think, probably the most exciting space in medicine there is. I
think so.
Because this idea, I mean, we always hear you are what you eat.
100%.
Never has it been more true than what science is showing us right now.
And I think it's really what made those 2 cases was really the donor, right?
Okay.
So, but other doctors have not been able to reproduce these cases.
They can't
right?
reproduce it.
So
Interesting.
what is it in the donor? So that's my question, right? What is it in the donor that allowed the microbiome to grow hair in those 2 patients, but not in others, right?
Right.
So I had a case of Alzheimer's where the husband lost his memory, and he had C. diff, and I did fecal transplant on him, and he remembered his daughter's date of birth after 6 months. So this was obviously the first that made us start looking, wait a minute, if we fix the gut, maybe we can fix the brain, right?
Wow.
But until so the problem is in medicine, people are in a rush to develop these products, right? Because it's all about the products. Well, it's not about understanding the diagnostic. We need the diagnostic. Imagine if people were bleeding.
Well, where's that? Is that rush from the investors or the
Yes.
parent companies that, look, I don't care how it works, it's working, let's get it on the market, somebody else will worry about that later, just hand it over. I mean, it's sort of what it is.
Exactly that.
Who cares why it works, just get it out there.
It's the story.
Yeah.
It's the story that drives the marketing, that drives the funds, and the story from the hope, right?
Right.
So you have a disease, you have Parkinson's, you have Alzheimer's, you want this to happen for you.
Hope sells.
Hope sells, right. But at the same time, there is hope with let's do meticulous science, and there is hope with let's just put mud on the wall and expect it to stick.
Right.
And the problem is if it doesn't stick, then they're going to go back and say, well, the hypothesis was wrong. There was nothing in the gut. But that doesn't mean there was nothing in the gut. It's just you rushed to the wrong microbiome implanted. You rushed the science. And that's not how science works,
right.
Unfortunately.
Yeah.
So with hope comes you have to do things meticulously. You have to have a good diagnostic tool. And so that's what we don't have right now is a good diagnostic tool. So as we are heading into a world of putting stools into the colon of people, we are going to have complications. We're going to have complications with, you swab C. diff for bipolar disorder because the person who is the donor, he's getting his $200 for donating poop. He is not going to put on the questionnaire, no psychiatric problem. He is not going to put on the questionnaire, didn't travel. Half the kids that are donating from MIT, they put on these questionnaires, never traveled, never used drugs, healthy. I mean, come on, right. Don't use alcohol. I mean, you're a college kid, okay?
Right.
Don't tell me. So that's the problem. The problem is we are trusting these questionnaires,
as you're saying,
people
this
that are
is
being
taking
paid.
something that has trillions of ingredients in
Yes.
it. Some of those ingredients we understand, most we don't. You take it over, in the one hand it had what you needed, but it could totally not have exactly what you needed or be out of balance. Clearly there's got to be a balanced conversation that happens here.
Absolutely.
How are they interacting with each other that's giving this
Right.
result?
And most people don't even know what the balance is. In fact, I read data and I read papers all the time on the microbiome and I'm like, oh my God, they are so wrong. So, this is the problem. We're following, one science, one group of people, etc. So, this is, unfortunately, if we don't do things meticulously, we're never going to have answers.
So let's get back. COVID's sweeping the world.
Yes.
And you start looking at treatment, like I want to
Yes.
get involved. So what part of the treatment conversation did you get involved in?
So
Are there
the
treatments
first-
that work?
Yeah, so first of all, the first thing I was doing is I was treating on the front line. So remember March 15, I wrote a protocol to the
Okay.
FDA. And I said, and I looked at all the data. So one of the data that I saw was from Dr. Didier Raoult.
Right, out of
And-
France.
Out of France.
Yep.
And what impressed me with Dr. Raoult was the way that he did the studies, the testing the PCR on a weekly basis. And by the way, remember, it didn't come to America yet when he was
Right.
already publishing his 20 patients.
He was already working people falling in China.
Yeah,
I think he had some
and
stories on there.
I speak French, so I got to read his first paper. So that was the first thing, is I got to read what are other doctors doing. And also I'm in the clinical trial business, so I have about, thousands of doctors that do clinical trials and that are on the front line of research. So I would make phone calls. I made phone calls to Italy. I said, what's working there? So Italian doctors would tell me vitamin C. I'm like, oh, vitamin C. And then I made phone calls to China, Hong Kong. I called some of my colleagues and I'm like, what's working there? Vitamin D. I'm like, vitamin D. And then I started looking at the whole zinc blocking COVID, right? And then I saw the hydroxy, Z-Pak, and zinc from Dr. Raoult. And I said, what impressed me was his methodology of his research, but also the fact that here he was risking his life, on the front line, and he was still surviving.
Right.
And remember, at the first few cases that we had of COVID, so here I am, I'm writing the protocols, ran it through the FDA. The FDA, within 24 hours, said, Dr. Hazan, move this protocol to full on. You're allowed
So
to
your
run the protocol.
protocol was what? Those things you said?
Hydroxy, Z-Pak, vitamin C, vitamin D, and zinc.
Okay, and the FDA heard that from you and said, great, we're in. Proceed.
Proceed with research, proceed. I didn't even need to do a clinical trial on it, okay?
Wow.
So then within 24 hours, I'm sorry, Dr. Hazan, we have to do a full-on clinical trial on this. So I said, okay, fine, I'm ready. So I said, well, instead of a phase 1, so clinical trials, there's different phases.
Okay, yeah.
Phase 1 is like, first there's the animal studies.
Right.
Okay, then there's the phase 1, then phase 2, phase 3, phase 4 sometimes, and then off the market, okay? So I said, can we skip the phase 1 because it's a safe drug? I mean, it's hydroxy and Z-Pak.
Right.
You know, it's
We use
not
it
that.
all the time.
We use hydroxy for lupus patients and Z-Pak. I mean, how many people have like the cold or flu and they give Z-Pak?
Right.
So I said, can we just move to phase 2? So
Is
they
phase
allowed
1 mostly like the safety side? Like
safety,
just make sure that this is
safety,
ethically
yes.
safe to move forward
Safety.
with? Okay,
Yeah,
got
it's
it.
a safety phase.
Got it.
So I said, can we just go to the safety and efficacy? Now remember, I'm still, I'm still innocent in the whole world of FDA and pharma and all this, and I'm thinking, I'm going to start my own pharmaceutical company, right?
Right.
Because I have this protocol. I patented. That was the first thing I did because I already was treating people. So I showed.
Where's Zelenko in this? Is this Zelenko ahead of you?
No, I had written the protocol. I had submitted to the FDA. April 3rd is when it went on clinicaltrials.gov.
Okay.
So we were number 3 or 4 on clinicaltrials.gov. So I had two protocols. One was the prophylaxis protocol, hydroxy, 2 pills only, and then vitamin C and D and zinc for 3 months.
Yeah.
So that was my prophylaxis idea. Why did I think 2 pills of hydroxychloroquine when you're exposed to someone with COVID? Because I felt, look, when you have a patient with scabies or lice, right?
Yeah.
You're going to treat you and you're going to treat the family, right? So the first thing is you're exposed, your wife just coughed on you and you have COVID. You take those 2 hydroxy, people don't realize that hydroxychloroquine stays in your system for 29 days. Okay. The half-life of hydroxychloroquine is 29 days. So therefore, to me, I felt if you're exposed, it's like I'm taking the pill.
Yeah.
I'm covered for 29 days. Now I just got to boost my microbiome with vitamin C, vitamin D, and zinc for the next 3 months. So that was the thought process of this protocol. And I started recruiting all the physicians at the beginning, okay? At the same time, so this was on clinicaltrials.gov. For those who don't know what clinicaltrials.gov, that is the site where every doctor who wants to know what's current in medicine or science goes to. Mm-hmm. You want to know what is the latest treatment for acne, you go to clinicaltrials.gov. Right. So everybody, it's the biggest platform in a way to raise awareness. And so what did I do when I posted it on April 3rd and 4th, is I sounded the alarm. I basically, in full transparency, said, this is what I believe as a pharmaceutical company, Topelia Therapeutics, Topelia, the genus for dove, for peace.
Okay.
I said, this is what I believe is how to prophylax yourself. 2 pills if you're exposed, and then vitamin C, D, and zinc the rest of the pandemic. And then the treatment, hydroxy, Z-Pak, vitamin C, D, and zinc, right? So I got approved. Then we had to go with what's called the regulatory body, IRB. So the IRB waited a month. We're in the middle of a pandemic, and a month. And by the way, my experience with that IRB was that previously, when I was doing pharmaceutical trials, I would give a protocol to the IRB. Within a day or a week, I would get that protocol.
Everything else you'd always done takes like a day.
Yeah, everything.
Suddenly, they're telling us we're in a pandemic. We don't understand this thing at all.
And
we're terrified. We need answers.
Yes.
30 days they're waiting to just give you an approval of something that always said, looks pretty good, go ahead.
And so I had to fight every day because I was, what do you mean? We're in the middle of a pandemic. How long is it going to take? People are dying. There's this. So I wasn't going to wait for my protocol. I was treating as a physician on the front line, and I started gathering my peers, right? So, and I started telling them about my protocol because I needed them to refer patients. Because what was happening at the beginning was we couldn't even advertise. So here we are, we've waited 30 days for the IRB to approve us. The IRB now says, okay, you're approved. That night, a tweet goes off. Dr. Hazan is doing unethical trials. Dr. Hazan, five drugs in a one arm. In other words, I wasn't doing a placebo arm, right? Because the protocol was approved as a open label.
Yeah.
Let's see if it works. Let's see if it kills people.
Right.
And then let's worry about bringing on a placebo or sugar pill, right?
Right. So
that tweet went off, and I really didn't pay attention to it, right? Because I said, who's paying attention to Twitter at this point? But it was sent by someone that works in public health at Oxford University that had 396 followers. So I started, and Oxford, remember, is the one that started Moderna vaccine, right? So I'm following the tweet, and it has 77 retweets, and my name is trashed already, and what's going on? And that's basically when, that was sort of like the first, red flag for me.
Right.
The next day, there's a Stat News, Dr. Sabine Hazan, a clinical trial company, Progena Biome, is doing a trial without placebo on 5 drugs. Is this even research? What are we doing here? This is totally not research. So all of a sudden, but that post didn't make it to mainstream media. That post made it to all the regulatory officers, the IRBs, and the FDA. That day, the FDA calls me and says, Dr. Hazan, I'm sorry, we have to ask that you do a placebo-controlled trial. Now, at the same time, Zelenko was like doing his own thing.
Yeah.
The doctors were treating. I was treating. So everybody was kind of on this rumble of hydroxy and Z-Pak, right? So, and by the way, I partnered with Dr. Tom Barodi on these protocols because Dr. Barodi is the father of fecal transplant. He's the one that brought triple therapy for H. pylori to market. And he's done tremendous work in medicine and in GI. So I felt, older physicians, scientists, that all of us as gastroenterologists followed because he really led us to the world of fecal transplant. So to me, I felt like this is the most legit physician that I could be part of with this. Yeah. And as I was running hydroxy, he was looking up ivermectin, doxycycline, and he was telling me, Sabine, it's all about ivermectin. It's all about ivermectin. So I said, Dr. Barodi, I'm too busy right now fighting with the IRB and at the same time saving lives. And at the same time, I convinced my scientists that we needed to look at the stools for COVID. So my scientist, Dr. Papuzzi, said, you're wasting, it's going to cost you close to $200,000. You're wasting your money. You're never going to find COVID in the stools. So I said, well, if I'm not looking at it, I'm not going to find it. So we basically had this little bet, him and I. And then the first thing I did, as I was writing the protocol, was also collect stools of patients with COVID. So the first patients that I was treating, one of them was like a pediatrician's, her daughter had COVID. A couple of friends of mine in Malibu or in Beverly Hills. And I started collecting. I'm like, I need your stools. I need your stools. So we had the first samples, like 8 or 9 samples. And so we had to develop a whole pipeline to see COVID in the stools. So that was the first moment that we discovered COVID was in the stools, whole genome, not a small little portion, but a whole entire genome, not one time in the stools, but thousands of times. In other words, thousands of copies of COVID in the gut.
All right, so let me just, there's this conversation going on a lot with our audience that it's never been isolated, that COVID was never isolated. Is that an accurate statement? Because it sounds like you're looking at something.
Yeah, so you have to remember this virus, and now the whole data is kind of, more in the open where we know it was manipulated, right? Yep. And I'm going to be upfront with that because I'm in the genetic sequencing business. In a way, and the reason so you have all these older scientists that are from an older mantra, right? Of, you have to isolate the virus. You have to give it, and then by reproducing it, then In other words, I isolate COVID, I give it to a mouse, the mouse gets COVID, there's my experiment, that's scientific. That's what we call in science, that's the old method of doing science, right?
Okay.
The idea of culturing a bacteria or a virus, right?
Yeah.
To therefore prove its existence.
Right.
We no longer have to do that now because we are in the level of genetic sequencing. So in other words, we look at sequences, codes, DNA, RNA, DNA then reproducing to RNA, and those sequences determine what is matched to the virus or to the
So
bacteria.
you're able to sort of using computer technologies get deep
Yes.
into this. You can isolate, pull this out, and see an entire genetic code and say, wait a
Correct.
minute, we know that's coronavirus because it has all the attributes of the coronavirus. We can see where, because I'm hearing all about these inserts,
Correct.
we see all of
Correct.
those things. And what you're saying is that for you, it doesn't make sense to going back and sort of scraping it, put it in petri dish and sitting around to
Yes.
see it, you know, if it grows or put it in a mouse.
Correct.
We can see, we have enough scientific evidence and we're so deep in, why go back to sort of an archaic isolation
ways.
process?
Yes. So, and
Okay.
you have the clinical, right? In other words, you have a patient that fits the PCR, positive PCR, even though these PCRs, you know, are so, the sensitivity and specificity is really poor in my
Yeah.
opinion. You still have a positive PCR, the same symptoms, right? Loss of smell, loss of nose, coughing, short of breath, fever, diarrhea, you know, the same symptoms. And now you also see in the stools the sequence of the virus, right? Now, if we didn't see the sequence of the virus and we had all these symptoms, I would say, well, you know, there's maybe there's no COVID, right? But the fact that we saw this sequence, call it whatever you want to call it, this sequence matching the symptoms, you have to put two and two together,
Let me
right?
just tell everyone that's not static on our microphone. We have a major rainstorm pouring
I know.
down on our studio here if you're wondering what's happening. Okay. So you see the sequence. You see if they have these symptoms. If I look for it, look, there's a sequence.
Correct.
If you don't have the sequence, the symptoms, I don't see the sequence. So you're sort of able to isolate through
Yes.
groups like there is these two things are going together.
Correct. And it removes this whole sensitivity and specificity because this is forensic medicine. You go deep enough into the microbiome that you find, and in a way it's in isolation. So we were the first lab in the world to use the technology to isolate the whole genome in the stool samples.
Okay, here's your study that you got into detection of SARS-CoV-2 from patient fecal samples by whole genome sequencing. All right, and so super fascinating. You find it. You see the
Yes.
work that you're doing work around it.
Now the government
Yeah.
knew about my work because I'm in contact. So remember, 4 years prior, I'm working with the government. I'm trying to standardize my lab test. I'm trying to do everything by the book, right? So I work with the NIH, I work with the FDA, I work with the NIST, National Institute of Standards, which basically standardizes all these tests.
Okay.
So I called the National Institute of Standards and I basically said, you know what, you guys need to pay attention in the stools because we're finding it. And actually China had found it, but by PCR, but they didn't find the whole genome sequence. So we said we found the whole genome sequencing in 100% of the positive patients, but also we're finding it in the asymptomatic. And when you take the history, you realize a week prior they had symptoms, right? So it was all correlating. It was like, basically forensic. So that's when the National Institute of Standards started looking at the septic tanks because they realized maybe that's how we track the virus. And that's how they started looking at the dorms and the colleges because now it gave them a new way.
So you set that off. You were the one that said you
yes.
can find it
In the stools.
In
yes.
the stools.
That was my whole platform because I felt, here, and it was so funny because you were talking about God before. This whole thing has been divine intervention for me.
Yeah.
Because it's the moment you realize you have a genetic sequencing lab, you have a clinical research company that does clinical trials, and both those things are really needed for this pandemic. And who else has both those companies except me, right?
Right.
And so I said to myself, that's why I kind of jumped, and I knew in my gut I was going to find COVID in the stools. And so when we found COVID in the stools, that's basically, we started noticing hydroxychloroquine and Z-Pak eradicated the virus in the stools. And so, 10, 12 patients later, I said, this is the way.
So you're proving a mechanism. I can use this, I can show you and prove the mechanism.
So I had the diagnostic.
Yeah.
I had isolated, if you want to call it isolated, I had found COVID in the stools. I had found that hydroxy and Z-Pak eradicated. So to me, that became my patent, right? So, and that became my clinical trial, right? What happened is that, and going back to the IRB, finally the IRB approved us. We get stopped by the FDA to run a placebo-controlled trial. So I convinced the FDA to let me do the trial, and the placebo was going to be high dosages of vitamins. So we basically, and I had to make my own vitamins. So I called a pharmacist in Beverly Hills and I said, formulate, I need a formula. I cannot have, the capsule needs to be vegetable, it needs to be pure vitamin C, pure vitamin D, zinc, because I need to give all these patients my vitamins that are in the clinical trial. I can't have them buy at, vitamins at CVS, Walmart.
So
I don't know
let me
the
get
quality.
this straight. To isolate hydroxychloroquine, you gave just the vitamin part? Is that what you're doing?
No, I gave hydroxy Z-Pak and vitamin C, D, and zinc.
Okay.
But the hydroxy Z-Pak killed the virus.
Right.
The vitamin C, D, and zinc, what we discovered as, we were testing patients with vitamin C on their own, vitamin D, it actually increased a bacteria called bifidobacteria.
Okay.
So that's where my path took me. So, but before I get to that, because I know I tend to be a bit hurricane-y,
That's
I'm
all
sorry.
right.
The IRB finally approved us. The FDA stopped us. IRB stops us again. And then the IRB comes back and says, I'm so sorry, Dr. Hazan. So this is May. They're like, I'm so sorry, Dr. Hazan, but we can't do your trial. You're going to have to find another regulatory board. That regulatory board was hired by Moderna to run their clinical trial, okay? So while they said that, I'm finding another IRB, and I'm trying to find someone that's preferably independent, small, boutique. It's not very easy in the middle of a pandemic. And I found someone, and then within a week, they approved the protocol. That week, or within that week, that's when, as I was open for enrollment, ready with my protocol, and remember, I'm in the clinical trial business. I know how to recruit patients.
Yeah.
I have my ways, from search engine optimization to everything, right, to get patients. I could not recruit one patient in that clinical trial because the lobbyists used they used Trump to create chaos within my protocol. So here you have Zelenko talking about hydroxychloroquine in hopes to save people. And I didn't really say anything back then to say, hey, it's my protocol, my patent, because I felt, he's saving lives.
Yeah.
That's what he's supposed to do. He's going to do what he's going to do. The guy had terminal lung cancer, and he was fighting this for humanity. So I said, this is his platform, this is his time, right? From Zelenko, the Trump, I was shocked because I live in Malibu. I'm trying to recruit in Malibu, my colleagues, and, nobody wanted to, they called it the Trump drug. So that protocol was destroyed. We could not even post an ad that is approved by the IRB on Facebook or Instagram. In fact, I'm banned from Facebook and Instagram on all my pages. I have like LA clinical trials, Ventura clinical trials. I have a lot of clinical trials pages to recruit these patients. None of them we could advertise. So that's when I started seeing the power of lobbying in America, right? And
so the Moderna vaccine lobbyists, the Pfizer, Moderna, probably Johnson & Johnson, all just go in and really just put pressure on these IRBs and the entire system to not let any other product. Now, and I've said this in many talks because I think the motivation is the emergency use authorization says you're not going to be able to rush the vaccine product out, because rush it through its trials if there's any other product that works. They can't have any other product work.
Well, if they had a product work, they would not be able to advance the protocol.
Right.
So they would not be able to advance the vaccines.
Right.
So I knew at that point, well, I'm kind of stuck with hydroxy. I'm not gonna, and I was treating people with it, and I was still looking at the stools, right? Because science is an evolving, tool, or an evolving field rather. And so in July, because I was stuck, recruiting, Dr. Barodi said, why don't we move to ivermectin doxycycline?
Right.
So that was July 2020. So July 2020, I started rewriting another protocol. And remember, these protocols are like 300 pages, you know, and you have to write a consent, and you have to, you know, it's a whole team 24/7, just rewriting, correcting, and then we have to resubmit it to the FDA, fight with the FDA to get it approved, then get the IRB to approve it. So we started in July. We got approved end of August for ivermectin, doxycycline, zinc, and vitamin C and vitamin D, because I believe that vitamin C and vitamin D and zinc is huge in the in treatment, prophylaxing, improving your bifidobacteria, improving your gut. So to me, that was the basis. And then so what happened was, you know, we passed through the IRB, no problem. We started, and then, you know, my friends and colleagues, remember back then, so it was Peter McCullough was on the hydroxypan.
Yeah.
And Pierre Corey was on the ivermectin.
Yeah.
So I basically called Pierre, and I'm like, Pierre, dude, there's a role for hydroxychloroquine too. And, you know, ivermectin is not just on its own, it's got to have other drugs. So I basically connected those two, because I said, we're at war here, okay?
Yeah.
Obviously, so somewhere along the line, and I told Dr. Barodi this, I said, look, I'm going to put these protocols to going, but I'm telling you, there's going to be another movement to kill ivermectin. And I was right, because, and that really ticked me when I saw that, because it was Seth Meyers, the comedian.
Yeah.
Yeah.
Seth,
Posted, a horse paste. And,
Right.
and the whole time I'm like, why is a comedian talking about ivermectin? He doesn't even understand. It's a fermented product of a bacteria that is similar to bifidobacteria. You know, so to me, this whole thing, like just little by little, I just, you know, my fumes started going on in this pandemic, because here I've treated everyone. No one died on my shift. No one. I mean, we're talking about, you know, officials, politicians, celebrities. I mean, I'd be on the front cover. You know, I'm a Malibu physician, right? So, and of course, nobody's talking because they don't want to talk about hydroxychloroquine. And they don't want to be political because their show is sponsored by pharmaceutical companies, right? So I was stuck in this little thing, right? Of here I am doing the research, here I am doing the clinical trials. I saw what was happening to my colleagues, right? The defamed, the defamation, the physicians
Which I'm
that were
still sitting here thinking, you didn't get sort of that free negative advertising, which these guys did. I mean, that's when I started looking, we started talking, I was like, wait a minute, you know all these guys, you're in the middle of it. In fact, you're in the lab doing the most important work, which is you can prove this.
Right. Yeah, and actually, you know, I tried to stay under the radar.
You
I
did
was
a pretty
letting
good job.
those guys. Yeah, thank you. Well, I think I was trying to stay under the radar, but I think there's also a huge force trying to keep me under the radar, because let's face it, I mean, you know, I've done over 300 clinical trials for pharma. So, you know, there's a lot of charts in my storage facility that we have to store for 25 years. So, you know, I'm the person that basically brought drugs to market, right?
Yeah.
So I'm, you know, in bed with, you know, the evil side, right? But unfortunately, there is, you know, and I'm not going to trash pharma, because pharma does do a good job, some pharma. But what we've seen in this pandemic is a rush of products, a rush of vaccination of everyone, and not proper research, okay? Because proper research would have been, let's do the animal. I understand rushing, and you're panicking, and you're this, you know, housewife that's 300 pounds, and you're afraid of COVID killing you, and you want a vaccine. I get it, okay? But at the same time, why didn't we continue the animal studies? Why didn't we continue looking at the genetics?
Right.
Is the vaccine affecting the genetics? Is it affecting the P53? We have the technology. Before and after, you could look at the P53 and see, did it affect it? Did that spike?
What is P53?
P53 is basically a marker that we test for cancer.
Okay.
So, you know, we have the technology. If we have a hypothesis and we say, well, these vaccines are causing cancer, right? Well, we can look at it and say, is it causing cancer, right? So basically, as all these protocols were going, I was doing it the right way, with the FDA watching, with the FDA coming to my office. And at the same time, I was also running other protocols, autism, fecal transplant. I was running, other protocols for pharmaceutical companies at the same time. So to me, because my whole attitude of why I stayed in clinical trials is because I am one of those doctors that is actually has an oversight. PI oversight is what I am. So in other words, if a pharmaceutical company comes to me and says, we want you to investigate this product, no problem. But if the product has a side effect, I'm going to be making the phone call to the FDA and saying, this is a side effect. I'm not going to hush it and close it under the, I'm not going to put it under the table. I'm going to report it.
Right.
Because that's what's called vigilance in research and in medicine. So that's why I stayed independent. People have wanted to buy my research center, to own me. I never wanted to. I want to do research the right way, because at some point I'm going to be the patient, right? I'm going to be the patient that's going to be taking that drug, and I'm going to be the patient that's going to be needing that drug. So we really need research to be done properly, ethically, and not, mess it up because of a price of a stock, because that's what it's all about.
So then, so ivermectin is getting killed, hydroxychloroquine, you can't get your trials done the way you want, so you decide, well, hold on a second, here comes the vaccine. Now the vaccine's coming out, your friends decide, most of medicine is going to jump in, and so you said, can I test your, can I get some fecal samples ahead of time and after? What did you think you were looking for? What was it that you wanted to see?
I honestly didn't know what I was looking for, right? I knew what the microbiome looked like, before COVID. I knew that in kids, babies especially, they had a lot of this bacteria called bifidobacteria.
Yeah.
I knew that, the probiotic industry, if you turn around the bottle and the label, it says bifidobacteria.
It's very important.
So, that was basically bifidobacteria was very important, right? So the first thing I started looking at is, what is the microbiome of people that have severe COVID versus people that have been exposed to COVID and never had COVID? Okay, so an example, a farmer. A farmer, his wife had COVID. He never had COVID. He took her saliva, smeared it on his face, and we did a YouTube video together, by the way. The YouTube was removed in the last couple weeks. And all we were talking about is farming and how it's similar to the microbiome. We learned so much about the microbiome from farming, fertilizers, poop, etc. So we were talking about
the biome itself. And
the biome.
Same thing. Right.
That's when you use a fertilizer, it's the poop of the cow. I mean, that's your microbiome, right? So we were talking about just all those things, right? And one of the things that he said that to me. So on that video, that was in 2020, I think, 2021, he said to me, he never got it. So the first thing I said is, Matt, on YouTube, I said, Matt, can I have your stools? And can I have your wife's stools? So what did I realize with Matt is what I realized with I did a study of 70 patients. 43 had severe COVID and 20 had exposure to COVID. And a couple, like 10 had, or 10 to 20, I can't remember, but it was 72 patients altogether. And basically what happened was the asymptomatic had bifidobacteria. The symptomatic had zero bifidobacteria. The exposed had a lot of bifidobacteria. But they also had another bacteria called Faecalibacterium prausnitzii, which we don't hear too much about it because it's not a pharmaceutical or probiotic. But this is another bacteria that actually is in the colon of people. Some people have a lot of it. Some don't have a lot of it. So those people that were severe with COVID had zero Faecalibacterium, zero bifidobacteria, high Bacteroides, which is a bacteria that's like not necessarily a good bacteria if it's out of balance, and then loss of diversity. In other words, their microbiome was eaten by something, right? So the hypothesis became, did the virus cause the dysbiosis, the imbalance, the killing of these microbes? Or did the killing of these microbes occur because of the fact that the patient started off with a messed up microbiome.
Right.
You know, you think of your patient that took antibiotics for a tooth and then all of a sudden, you know, gets COVID. Or the person that's like drinking heavily and the next day gets COVID, right? He was in a big crowd, drank heavily. Did he kill his microbiome and therefore was he exposed to COVID? So that was really the beginning, right? That showed me this is what I may be looking at in my colleagues with the vaccine. So I really wasn't looking at, hey, let me pinpoint the bifidobacteria specifically. I was just seeing what is a severe
There's
COVID.
a pattern you're seeing.
Yeah, I'm seeing a pattern with severe COVID. I'm seeing a pattern with highly exposed COVID patients. And then let me see, I didn't even know. And honestly, you know, I wasn't, you know, I was not convinced of this vaccine from the beginning because of the fact that I saw the mutations. Remember, those 8 patients, they were all different. They all had a different mutation. So some people had like 4 spike mutations, some people had 3, some people had 1. So, you know, there's different combinations. And as you see the progression of this virus, you see it has 1 head, 1 body, 1 tail. And then you start seeing it with like 10 heads, 10 bodies, 10 tails, and then 20, 33. So there's multiple combinations to this virus. So when you get a vaccine, and I'm giving you a spike protein, right? They tried to mimic that original spike protein from Wuhan, right?
So the 1 head, 1 body, 1 tail, and now it's already got 20
Well, they took just
heads.
the 1 body, the spike,
true,
right?
Right.
So now we're at 33 spikes. But there's different mutations. So you're going to put 33
Just doesn't make
spikes.
sense
It doesn't make sense. So you're always catching your tail trying to beat up this virus. So to me, the idea was build up the microbiome, right? Let's look at the microbiome. So that's when I started looking at so as I was stopped, you know, from recruiting and, you know, and also I wasn't doing too much clinical trials. I had a lot of time, well, sort of. I do a lot more than most people do in one day. So I basically...
I can get that. I get that.
so for me, it was like, okay, hurricane mode, let's get stools. So I started collecting stools from my colleagues and, you know, and people would come to me and say, hey, I heard you're doing a trial on looking at the vaccine before and after. And I'd like to make sure that I know what my baseline microbiome looks like before I get vaccinated, right? Because we don't know what's in the shot and, you know, is there contamination? We don't know, right? There was so much we didn't know. So I basically, you know, I accumulated like 4 samples. And one thing that I noticed was the bifidobacteria, which is this important billion-dollar industry, drops by 50%, and some of them drop to 0
after vaccination,
after this vaccine. So then I said, okay, well, let me look at more people, right? And then we looked at 34 patients, and we noticed that indeed the bifidobacteria dropped. What was interesting is I didn't lose track of these patients, right? I basically said, okay, well, let me keep looking, right? Now, remember, all these stool samples are expensive. So I collected the stools to say, okay, well, let me continue looking, right? And what we discovered is that 90 days, right, into the virus, into the vaccine, the bifidobacteria dropped from like, you know, if you were at a million reads, for example, or relative abundance of maybe 5%, to 0 in 90 days. The damage kept on persisting.
Wow.
So basically, and that's what you're seeing, you know, and that's 90 days
So
and
you
then
saw
6
a drop
to 9 months later.
Wow. So you saw a drop up front, but a serious drop over time. It's getting worse. It wasn't correcting itself.
And in fact, what you saw with that paper, were 4 patients. And one of them was actually taking vitamins. So we thought, well, it's probably skewing. That's probably why he went up and he was doing probiotics, etc. But then, even though he was on the probiotics and the vitamins and everything, his bifidobacteria still dropped. So this is why I didn't really change anything in those patients. You know, I didn't change before and after because I wanted to see, I said, I want you to do the same diet. I want you to do the same. If you're taking probiotics, if you're taking vitamins, just stick to the same program because I just want to see
this vaccine doing by itself.
What the vaccine is doing by itself. And then what happened is, as I was seeing that, you know, there was a little girl and I'm not going to say her name, but paraplegic, vaccine injured, who everybody that listens to your podcast knows, came to me, and I decided, let me look at her microbiome, right? So I looked at her microbiome and her brother, who had gotten the placebo. Now, remember, kids have a lot of bifidobacteria, okay? This kid had 0 bifidobacteria. Wow. Her brother, who was basically got the placebo,
same trial, but in the placebo
had
group.
a lot of bifidobacteria. So here's in the same family, the kid that's paraplegic that got the vaccine got 0 bifidobacteria, and here's this.
I had someone I know recently, this is why it really actually got my interest, that got the vaccine, and then I would say weeks, if not a month or so later, told me, oh my God, I got the worst food poisoning of my life. I had to go to the hospital. I almost died. I've never seen anything like it. I think I got a piece of bad meat or something like that. And it just didn't really, there's something about it.
And it lingered, right?
Yeah, and they're really struggling to get back to feeling healthy again.
Yes.
Would that be sort of what we would see if you lost your bifidobacteria?
Yeah, that's the trend, right? That's what we're seeing. So I started seeing, Dr. Denise Hertz, who's been very, she's a gastroenterologist in Beverly Hills, has treated tons of celebrities, a prominent gastroenterologist, got vaccinated, and herself became debilitated after the vaccine. And she speaks about it. And I tested her stools because she actually came to me from a neurologist that said, hey, Sabine, I know. And by the way, all my colleagues thought I was crazy at the beginning because they're like, look, you're becoming an anti-vaxxer. And I'm like, I'm not an anti-vaxxer. I put these trials to market, right? I want to see the science. I want to see the data. It's not being an anti-vaxxer to ask questions and to defy the narrative. It's not. Anyways, all these doctors now believe me because guess what? They've seen CMV encephalitis. They've seen, herpes encephalitis. They've seen increase in Parkinson's, increase in Alzheimer's. They can't explain it. So this doctor came to me. And by the way, Dr. Denise Hertz is one of the most ethical, righteous physician, and we're very respected in the community of physicians, okay? And she came to me and was debilitated. I tested her microbiome, 0 bifidobacteria. And let me just say, from there we started doing, her and I started increasing the awareness because I'm not afraid to speak. I'm not afraid for full transparency, everything about my life. I'm on Twitter every day. I even say what I'm eating.
Is this the first time it's been done? Just because I know that this is something, this gut biome thing has come up in the work that I've done, people looking at autism. We've seen the fecal transplants there. I know you've been involved in some of that. But we really haven't ever looked at this element of what are vaccines, and I would guess drugs too. Do you think that as we move forward, it seems to me good science, especially as we're realizing how important this gut biome is, that a part of safety trials and efficacy trials should be testing stool samples before and after a product?
100%. And not only before and after, but 9 months, a year. We want to know, right? Same thing with antibiotics. Yes, we understand antibiotics kill strep pneumonia that you have because strep pneumonia is going to kill you at this moment. But is the antibiotic killing the rest of your microbiome, and therefore are you going to have a bigger problem down the road, right? Think about going back when we were doing antibiotics for everything, right?
Yeah.
Then C. diff started popping up, and then we had to do fecal transplant. It was no longer a pill to fix, it was taking stools from a healthy donor. What happens when you can no longer count on the healthy donors? And that's why, by the way, I started looking at...
I mean, imagine if you just with a vaccine wiped out
the whole population.
We could see that entire population.
And that was the mistake. And that's why I stood up, is because we are not equal in our microbiome. Some of us have a resilient microbiome, right? You think of the person that survives COVID and never got COVID, the whole pandemic. Resilience, right? You think of the person that got vaccinated 5 times and never got a side effect from the vaccine. Resilience. So resilience is an important thing that we need to protect, to preserve, because the resilient ones are going to be the ones helping the non-resilient. So if there is something to this, science of mine that I'm directing, and we're killing the microbiome, well, you have no hope for Alzheimer's. You have no hope for Parkinson's or autism if you're demolishing the entire microbiome of humanity and also globalizing. what makes us believe that we can just have a pill that fits all of us?
Crazy.
Right?
Yeah.
I mean, Africans have a different microbiome. They're in the jungle. You give an antibiotic to that African, you wipe out his whole diversity of the microbiome. You know, you give him antibiotic, or now what I'm showing, a messenger RNA to the Amazon jungle. They can't survive. They've adapted to living in the jungle because of their microbiome. So once you change that microbiome and you make them like city folks, they will not survive, you know. One little microbe that's in the earth will kill them.
We've gotten really deep into this, and I want to have you back on. But you showed me an image, and it's in sort of a different space, but I want to share it just to sort of close this out, because it really gives a good visual. We've got a slide, and my understanding is you have triplets and looking at autism.
Triplets and autism, so you could look at the slide.
Okay, let's take a look at this slide, folks. This is super interesting.
Well, this is actually my fecal transplant. So this is the case of fecal transplant.
Okay. Yeah.
So the first column is the child with a bacteria we've identified as Lactobacillus animalis.
So that giant blue is just
one big microbe just taking over the gut.
Taking over everything, totally out of balance.
Totally out of balance. He came to my office kicking his head, breaking his teeth. The parents, each one of them were taking turns at staying up all night to stay with him because he could not sleep. He had like a huge amount of proteobacteria, which are not necessarily your good microbes.
Okay.
The second column is his sister's microbiome. Now you could see she's a neurotypical child that is diverse, amazing. And then after that, after transplant, the kid matches his
Now, what did you
sister.
Transplant with? You take the sister's...
His stools. Her stools.
You took her stools, gave it to her brother who's out of
Via
balance.
colonoscopy.
And the third one is what happens to that biome. We get that blue under control.
Yeah. And then after and continuing, and you could see he started with a Shannon index of 2.2, which is essentially a loss of diversity.
Yeah.
And then he went up to 6.2. And I'm happy to say last week he went with a girl to prom.
Wow.
So this kid is sleeping. He's doing amazing. So there is hope, but we cannot kill the diverse microbiome. And we got to understand what is killing the microbiome, you know, and especially in this messenger RNA vaccines that are developing. So is the spike protein triggering other viruses and therefore killing the microbiome? Possibly. So,
well, I look forward to seeing more of your work. I want to thank you for taking the time.
Thank you.
So incredibly fascinating. Anything we can do to help, you know, if you ever need funding or help or support, that's what we're here for.
Thank you. Yes, we're trying to look at the microbiome of newborns, of vaccinated moms versus non-vaccinated. So
fascinating.
Please.
Do you have how do we follow what you're doing? Do you have a foundation?
So
or any way
yeah,
we
the
can
Microbiome Research Foundation.
The Microbiome Research Foundation. Here we go. MicrobiomeResearchFoundation.org, folks. I think we need to get involved in this process. And you can even sign up for clinical trials.
You can sign up for clinical trials. We basically, on Progena Biome, we have a bunch. We put your name on a list, and as we get funds and as we need patients, you know, we're doing Parkinson's right now. We're looking at the microbiome of Parkinson's.
Wow.
And then from there, we're going to do animal studies to see if those microbes are real and create a mouse model for Parkinson's. So a lot of exciting things. I'm working with Dr. Adams. We're trying to develop an assay for autism so we could prevent autism, but also try to treat it by what you saw with the technology. So, you know, there is hope in the future. I think we cannot censor. I was very disappointed to see that one of my papers on Frontiers, I found out Frontiers journal is a predatory journal.
Yeah.
I published the paper. It was a hypothesis on why ivermectin works.
Yeah, do we have that? I think we could just pop that up really quick.
Yeah, and that was
and you said it's a hypothesis. It's a hypothesis of ivermectin affecting the biome.
Yes, and
microbiome-based hypothesis on ivermectin's mechanism in COVID-19. Ivermectin feeds bifidobacteria to boost immunity. It says right there it's a hypothesis. Why would they get pulled? The whole idea is let's start a discussion. Anyone else want to jump in and take a look at this with me?
You know, science is about prove me wrong or prove me right.
Now it's about censor you, shut it down, burn it, make sure no one ever sees it again.
And by the way, I sent that hypothesis to every physician I know in academia so they could see what's going on in the medical field. So 'cause I'm not going to let this slide.
Amazing. You are a superhero.
Thank you.
It's really great to finally have you in studio. We look forward to seeing more of the great work that you're doing. When we, look at this entire cover-up and who was involved, and you think of the media, how you watch, Seth Meyers, what is a comedian doing, saying things that is destroying studies that are trying to be done? Yes, Seth, you had that amount of power. Maybe we put you on the list that got people killed when we look back historically at this whole thing. But somebody took task with the BBC, someone you might know, on almost exactly this issue. Take a look at this.
You've changed the COVID misinformation
Has
rules.
BBC changed its COVID misinformation?
The BBC does not set the rules on Twitter, so I'm asking you.
No, I'm talking about the BBC's misinformation about COVID.
I'm literally asking you about, you changed the labels, the COVID misinformation labels. There used to be a policy, it then disappeared. Why do that?
Well, COVID is no longer an issue. Does the BBC hold itself at all responsible for misinformation regarding masking and side effects of vaccinations, and not reporting on that at all? And what about the fact that the BBC was put under pressure by the British government to change its editorial policy? Are you aware of that?
This is not an interview about the BBC.
Oh, you thought it wasn't?
I see now why you've done Twitter Spaces. I am not a representative of the BBC's editorial policy. I want to make that clear. Let's talk about something else. If you want to talk about the BBC, all right. Let's talk about something else.
First of all, I just want to say, Elon, you're on my bucket list, man. I'm here in Austin. If you ever find yourself sweeping through town, we would love to have a conversation with you. I've got some serious questions about a lot of what you're doing, but also so excited about, I think you've been one of the biggest news stories, but here at the High Wire, we've gotten so much of this right. We didn't listen to the mainstream. No one was pressuring us because we don't have sponsors. I'd love to have a conversation with you about how we all move forward in this really dangerous and precarious world. You have such an interesting voice, and you're just one of those people I think would be fascinating to have here. So next time you're in Austin, why don't you give us a call? All right, look, this is the High Wire. We are so excited about what we do here. I am so thankful that I'm a part of this network, this growing body of people. That's you out there that are actually making a difference in the world, that are actually stepping up and doing something, standing for science, standing for medicine, standing for freedom. And it doesn't matter if you're a mother or you're one of the great, microbiome specialists in the world. We all have a job to do right now. Our children, the future of this species is dependent on all of us breathing right now. If you're watching the High Wire, that means you. That means everyone you know. How many world changers are out there that just don't really know the truth yet because they're not looking in the right place? Please show the High Wire and get them focused in the right place so that we can inspire them, because we need them. That specialty, that thing they do that makes us all unique. We're all God's creatures. We are all a part of something so important. We are watching the world close in on us. The liars, the thieves, the moneyed interests are trying to rob our world from us and make decisions that could wipe out everything that makes us healthy, everything that makes us free. It has never been a more important time to be alive. There's never been a more fantastic and special time to be a human being. Celebrate what we're in right now. No human being has ever been needed more than you are needed right now. At least that's what I think. That's why I'm doing what I'm doing on the High Wire. And I'll see you next week.